Well, welcome everyone. Thank you for coming to the second Jones Seminar of 2015. It's a pleasure to have another semi-local speaker today, Sarah Moore, who's just down the road at Smith College. She's an assistant professor of engineering at Smith. For those of you who haven't been here for that long, Smith and Dartmouth have had a long relationship together in that Smith students used to come here to get their engineering degree. That was probably 10 years ago, and about 10 years ago they created their own engineering school, and since then, and Thayer was involved in giving some advice on how to set up an engineering school, and in fact our own dean of Thayer's school Smith today helping them do their 10-year evaluation. So there's been over the years many connections between Smith and Dartmouth, and Smith now, I'm told, is the only engineering program in a women's college in the United States. So that's been a big development and exciting to see. So Sarah is a recent addition to Smith, only been there for just over 2 years now. She graduated from Princeton University in 2006 with a BSE in the Department of Chemical Engineering. In 2008, she received her master's degree in bioengineering from Stanford in a department jointly housed in the School of Engineering and the School of Medicine. And then in 2012, she finished her PhD in bioengineering. Her graduate research focused on engineering novel peptides for applications of molecular imaging in cancer. And immediately after graduating from Stanford, she joined Smith College, where her group focuses on engineering molecules to serve as therapeutic and diagnostic roles in oncology and disease in the central nervous system. So, Sarah, thank you very much.
Thank you. It's great to be here. Have had some really interesting conversations throughout the day. Really also feel drawn to the way that you all approach both your education and research here, in the sense of it's really crossing boundaries where you don't feel siloed, as I am a mechanical engineer and I am an electrical engineer, but looking at important problems and thinking about what are the important questions you need to ask to help solve those problems. So thank you for inviting me today and for spending time with me today, including for this talk. So I'm going to talk to you today both about some recently published work as well as next directions that I'm currently pursuing with my lab in this general field of engineering proteins, engineering biological molecules for applications in human health. And specifically today I'm focusing on some of the cancer work that I've been doing. But I'm also interested in questions related to diseases of the central nervous system beyond oncology. And I'm happy to stay here afterwards and talk with anyone who wants to talk more about that. So just to I like to give some context. I know it's a broad audience, especially when we start talking about proteins. That's something that's very important to me as someone who engineers proteins. I want to orient you with where I'm talking about there just briefly. Right? So proteins have all these roles, these important roles in life's normal processes. So talking about protein, talking about the product of genes that are part of DNA. Right? And so I'm engineering proteins. This central dogma is really important in the sense that I'm engineering DNA to have mutations to create new proteins that don't exist in nature. But when you think about proteins that do exist in nature, they have a variety of very specific functions in the body. So shown here is hemoglobin, involved with transport of oxygen throughout the body. Antibodies are proteins involved in the immune system, right? This is a protein that I'm currently working with. It's a domain of human fibronectin, which creates structure for the extracellular matrix that your tissues adhere to. Communication, so signaling in the brain. And this is an enzyme that is called lactase. It digests lactose found in milk. And if you're missing the ability for the body to make lactase, you can take a pill to replace that lactase so you can have dairy products. So knowing that there's all these very specific roles that proteins can have in the body, that means that we can take that and make new proteins to have useful roles in diagnosing and treating disease. And so that's what my research is focused on. And one of those applications in particular that I'm interested in is engineering proteins for these applications in molecular imaging. So when I'm talking about molecular imaging, I'm talking about looking at the molecular scale processes that go on in the body. So rather than in traditional imaging, where you're looking at these larger structural changes or existences in the body, really understanding what's going on at the molecular level in health and in disease. And I know a lot of people here are working on that, so I've had some great conversations today to hear what you all are doing related to molecular imaging. So just kind of at one level of this, in molecular imaging, is a protein receptor in the cell membrane in disease. Maybe that protein receptor is turned on in cancer, and so if we can see that it's there, we know that cancer is there. So if we want to look for that and see that molecule, we need some molecular imaging probe, something that can bind to very specifically to that molecule that we're interested in detecting and observing. And then beyond just having the probe, we need some way that doesn't just bind, but then has some signal, some readout that we can use with various imaging modalities, just generically indicated here by a star. That could be a fluorophore if you're doing optical imaging. It could be a radioisotope if you're doing tomography, something that has magnetic properties if you're looking at MRI, something that allows you to then locate where this molecule is. And then if you inject that probe with that label somehow into a patient and it binds to that receptor, that molecular scale property that you want to detect, then you can locate where that receptor is. So in this example here, this is a molecular image, a PET image of a patient. All to the image here, so hands would be up. These are our lungs right here. And this bright spot here is showing where a lung tumor is. And in this example, there is a particular cancer receptor that's present in tumor tissue but not in healthy tissue. And so then you can locate this tumor. And usually you can do this before you'd be able to see the more gross anatomical changes that happen with tumors. So molecular changes, the time scale with which when this happens is much more rapid when tumors start to grow and also when they start to respond to therapy. So if you can use molecular imaging to look at tumors, you can get information earlier about the presence of the tumor and earlier about whether or not it's responding to therapy so you can make better treatment decisions. And so that's one of the big potentials for contributions of molecular imaging is earlier information allows for better patient outcomes. So we can use molecular imaging to improve our ways to study and understand cancer as well as diagnosis and treatment. And so I'm going to share just two examples, or two images here, of before and after treatment. So again, this is a patient. We're looking at liver here. This would be kind of a full body slice, so the spine down the middle. And this is being imaged with a molecular imaging agent that binds to a particular receptor that is just, it's endocrine receptor, so binding to estrogen receptor. And these are metastases that can be seen that are expressing this estrogen receptor. And this patient then received a therapy after diagnosing these tumors, saw that these tumors were indeed expressing this receptor, received a therapy that specifically targeted that receptor, and if we didn't know that patient had those molecules present, we wouldn't know to give them that therapy, right? So we gave them this therapy, matched it based on that presence, and then following response to therapy, imaged this patient again. So this isn't my work, but this publication here, and you can see that those tumors do actually disappear. You see the absence of those molecules indicating the absence of those tumors. And that's the type of information you can often get with molecular imaging you may not be able to get with traditional modes of imaging. I also, in addition to engineering proteins that can serve as molecular imaging agents or other diagnostic agents, the proteins that I'm engineering in my lab are also targeted at being therapies, so stopping that grow and divide process of cancer cells. So, for example, this cartoon here is a cancer cell with receptors receiving growth signals that are telling them to grow and divide beyond how they would normally be checked. If we can engineer some protein, some molecule that is able to specifically bind that receptor, displace our grow and divide signal, then we can stop the growth of cancer. So that kind of broadly, as far as background, those are some of the ways that protein engineering can contribute to detecting and treating tumors. So more specifically, I want to talk about some recently published work engineering a very specific peptide that we were able to use for optical imaging of these brain tumors, medulloblastoma. And I'm giving the kind of punchline end goal here of this work, or at least the punchline of the work to this point. These are 3 mice. This one has a brain tumor. These 2 don't. And in this red region here aligns with when we visually inspect this removed brain where the tumor tissue is. So little bit more story about how we got to that endpoint, or that midpoint really, because it's not an endpoint until it's in people. So medulloblastoma is, of pediatric brain tumors, the most common. This is an MRI of a 4-year-old patient that has gadolinium contrast, and this bright spot here is showing a region of enhanced contrast, which is a medulloblastoma tumor. So about golf ball size lump at the base of the brain here for this patient. So the current treatment regimen for this patient, or the current diagnostic regimen, is this patient will present, maybe the brain's been affected in various ways that might make them dizzy or having headaches, or might be affecting their speech, it might be affecting their motion. These general symptoms that have something to do with misfunctioning brain as that tumor grows and moves away the brain tissue in that region. And so they will receive an MRI. At some point, a physician may say, okay, let's get an MRI, something seems to be going on with the brain, and they'll look for this tumor. And so what this patient will currently receive is they'll have surgical resection of that tumor. So a neurosurgeon will go in, remove that tissue. It's the first step. And then they'll receive general radiation over the entire cranium and the spine. This kind of trying to focus that radiation hasn't been shown to improve patient outcomes at all, so it will be general. Remember, these are kids, they're growing. That removal of tissue, if there's any healthy tissue in there, and that general radiation are both very damaging to the child's development. And then some of these patients who receive general chemotherapeutics, toxins, they don't seem to help that much with improving patient outcomes, but there's not much choice sometimes if it's the patient has a very large tumor, to hoping they'll survive, so they'll be to try everything. So any opportunities that we have for better clinical practice to either more specifically detect these tumors, specifically for more complete, accurate surgical resection. So one of my collaborators on this project, her name is Melanie Hayden, and she's a neurosurgeon. These are her patients, right? And so when she is in there in a surgery, I've seen her do this work and seen videos of this work, it is messy in there, right? It is actually really hard to tell what is brain tumor tissue and what is surrounding healthy tissue. The tumor inflames the surrounding tissue, so the healthy tissue, which is normal healthy brain cells that you want that child to get to have so they can learn how to run and speak and read and all of these things, looks just like the tumor tissue. So if we're able to light up that tumor tissue in order to remove that tumor tissue without removing healthy tissue, then that will improve our clinical outcomes. We don't want to leave any of that tumor tissue because then it has the opportunity to grow back. And I know some of you here are doing some really important work in this area, also trying to look at these better surgical removals of tumors to really enable better outcomes. And building on that, if we are able to get molecules very specifically to tumors, which is something successful as a molecular imaging agent, that means you usually are getting it specifically there, then you have the opportunity also for specific therapeutics, for targeted therapies. Right. So in order to be able to do any sort of molecular imaging for this patient, using approaches in protein engineering molecular probes, you need first some marker, some biological marker that is present on tumor tissue but is absent from healthy tissue. And in a variety of tumors, one such molecule that represents that class of molecules includes integrin receptors. So these are cell surface proteins. They are often present in the vasculature of tumors as well as on the surface of the tumor cells themselves. And they are involved with growth of blood vessels as well as the growth and spread and metastasis of the tumor cells. So in this case, I gave this earlier generic example of a marker, this time I'm labeling it with these two subtypes, these two monomers. Integrins are heterodimers. They have a ligand binding site on their surface in the extracellular space, and then a cytosolic domain, once it's bound, will signal to this grow and divide process. And these are generally, so there's a bunch of subtypes, and some subset of those subtypes are generally turned on in development and then turned off during adult states and also even during early childhood states. These are turned on early, but then turned off. And then they get turned back on during tumorigenesis. And in a lot of the kind of early question that I'm asking when we're starting a new project is what interesting biomarkers are out there that have this behavior where they're present in tumors and doing something in tumors that's not going on in healthy tissue, because we need that difference of presence in order to be able to get molecules just to tumor tissue and not to binding healthy tissue that happens to have that same receptor, right? So in this case, integrin receptors are broadly expressed in a lot of tumors. Prior to this project, we weren't sure if they were expressed in medulloblastoma, but I was working with a molecule that was able to bind very specifically, and we engineered it to bind very specifically to integrin receptors. So this is a structure of a protein that I work with. It's referred to as a cysteine knot protein, or knotin. So shown here are 3 disulfide bonds. It's only about 30 amino acids, and it has these projecting loops that are able to bind to various targets. So natively, the protein that we started with is found in squash seeds. So it has nothing to do with binding tumors, nothing to do with integrin receptors. It's actually a trypsin inhibitor. So if you've done cell culture, you might have used trypsin, which is an enzyme that chews away at proteins. Right? So it's a trypsin inhibitor. So kind of what's hypothesized its role in squash seeds is if an animal eats a squash seed, this trypsin inhibitor molecule is there, and it allows the seed to pass through the digestive tract undigested, and so the squash seed can become a squash. Right? So that's not what we care about for cancer, but we do care about the fact that there's these loops that are composed of amino acids that if they are mutated, if they are changed away from their normal sequence involved in trypsin inhibition, that structure still maintains its fold, which means we can tweak those amino acids. So these are the one-letter representative code for the amino acids, to, in this case, put in this red loop, which does bind to integrins. There's some rational engineering in this work. Sometimes you know a native protein sequence. In this case, we had a starting point. This is a 3 amino acid sequence that binds some integrin receptors. But often you don't know as much as you would like to rationally design a protein to bind to a new target. And so that's where a lot of the work that I do in directed evolution comes in. So we have this protein that binds to this receptor, very high affinity, so sub-nanomolar binding dissociation constants. And how we got there is with this process of directed evolution using yeast cell surface display, which Margie Ackerman's lab also uses here. So developed in the Wittrup lab in 1997, and this is a directed evolution technology platform. So when I say directed evolution, what I'm referring to is going back to that DNA to RNA to protein. And in nature's evolution, mutations at the DNA level get carried into mutations at the protein level. And then there's some selective pressure, right, that makes some protein more fit for an organism. So if that protein makes you run away faster from a predator, then that person who survives, that tiger, is more likely to have offspring, and then that protein prevails with these new mutations. In protein engineering, what we're able to do is create some external selective pressure, in our case binding to a tumor receptor, to identify that bind very tightly to tumor receptors from tens of millions of variants, many of which have absolutely no binding to that receptor, but we can identify those that have that really tight binding, and those have interesting applications in human health. So in these surface display, we have plasmids that express, in this case, it was that cysteine knot knotin. In other work, I do other protein molecules on the surface of a yeast cell, where they are tethered to the yeast cell by a set of yeast cell wall mating proteins. And there's also tags for detection that are genetically fused to the end of this potential protein that might bind a tumor target. And so what we can do when this protein is in this context of on the cell surface of yeast, we can incubate that yeast cell, which has about 50,000 copies of a single protein, and tens of millions of other yeast cells that all have a slightly different variation of that protein on their surface, and incubate it with a fluorescently tagged target protein, in this case integrin receptors, other projects, other receptors that I'm interested in studying and detecting, and can use flow cytometry to incubate, if these are individual yeast cells, each expressing an individual unique protein, add the fluorescently labeled target protein. Some will light up with flow cytometry, and then we can collect those individual yeast cells and repeat this process to enrich for those clones, those proteins that bind our tumor target, and then be able to recover the DNA and identify what are the protein sequences that have the tightest binding affinity to our tumor target that we're interested in. So this is the process that we went through to identify these integrin binding proteins, these integrins being prevalent in a lot of different tumors that we might be interested in working with. Okay, so we have these patients, these medulloblastoma patients, who have a lot of needs for better detection and treatment of their tumors. And we had this protein that we've been working with in other contexts, in model systems that bind to integrin receptors. But really, when I ran into my friend Melanie, we were in labs next door to each other. She's saying, hey, do you have anything for my brain tumor patients? Started talking about, can these engineered proteins bind to integrin receptors specifically to bind to and identify medulloblastoma? So are those receptors present? If we're going to use this molecule in these medulloblastoma, that's an important first question. And as part of answering this work, Melanie had been developing these mouse models of medulloblastoma that accurately recapitulated how medulloblastoma develops in the human cases. So shown here is a mouse brain, and you can see the gross morphology of the tumor is different than this nice ordered structure we have on the other side of that brain. So this was a tumor model that she's developed, stereotactically injecting tumor cells into the mouse brains. And all these mice, almost at 100% take rate, will form tumors in about 3 weeks. She also has a genetic model that's also coupled to a fluorescent reporter, so these light up with GFP when a tumor forms. And this is a genetic model that has a mutation in PATCH, which is part of the hedgehog signaling pathway. And about 25% of human medulloblastomas also have very similar mutations in that pathway. And so these form in the genetically same way that 25% of human medulloblastoma tumors form. So also a very close genetic model of the human case of disease. A challenge with these tumors is only about 15% of these mice form tumors, and only after about 4 to 6 months. So if we're looking at wanting to do large enough numbers of animals to be able to study and get data on whether our probe is specifically medulloblastoma, this is a more difficult model to work with, but captures the human genetic progression. This model enables us to do a greater variety of studies, but isn't as closely matched. And so we worked with both of these models in this work. And so getting back to that question of, well, do medulloblastoma cells, due to these tumor cells express the integrin receptors? And that was the first question we asked. So they do. And shown here are just 2 different examples of some of the data that we looked at. These are flow cytometry plots here, where basically the red curves are negative control, and blue is looking for specific subtypes of that integrin. And these are cells, medulloblastoma cells, mouse medulloblastoma cells, and they do express the integrins. And confocal microscopy, blue is the nuclei, red are cell processing, and green is some of the integrin receptor for various types of integrin receptors. So the receptor's there. So we have this engineered protein, we have the receptor for these medulloblastoma cells. And beyond the cellular level, it's at the tissue level. So this is histology work. In this case, green are the tumor cells, because they're the GFP-expressing tumors. And red, we see both in vascular structures, as well as on cells, we see integrins present in the tumor vasculature and on the tumor cells. It's not just at the cellular level, but in the tissue level, and it's not present in healthy tissue. So quantitation here, looking at these 2 integrin subtypes, 1 is indicating in the tumor tissue, 2 is indicating outside of the tumor tissue. And we have that differential expression that we need. We want to specifically target tumor tissue, but ignore the healthy tissue. Okay. So we have the engineered protein that we've engineered with directed evolution to do something it doesn't do in normal activity, some new activity binding these integrin receptors. And we have this patient population that does have tumor cells that express this receptor. And I will say all this work was in mouse. Some follow-up work that I'm not including here was able to get patient clinical samples and show in a smaller subset that those could be transplanted in the mouse and still have a lot of this, still have the same results here. And they do express the integrin receptors, and so you always want that human validation, and they do have that. So then thinking about that star part of that molecular imaging probe, how we'd actually be able to see using some imaging modality. In this case, what we chose to work with is a near-infrared imaging probe, Alexaflor 680, and had chemistry to be able to couple this optical imaging label to the N-terminus of the protein that binds to integrin receptors. And just as a very specific site-specific coupling, this protein has no other chemical handles where this probe would attach with the chemistry that we used. And confirmed in cell binding assays that the unlabeled version compared to the labeled version both still bind cells. So our labeling with our probe or optical imaging probe aspect of this molecule did not interfere with that ability to bind to our integrin-expressing cells. So that's basically our in vitro test, right? So that's this protein can be labeled and can bind medulloblastoma cells that express integrins. And that's where often a lot of things then fall out. You do the in vitro work and it looks great. And then this works in vivo, right? So as I showed you at the beginning, this works in vivo. So we injected this near-infrared labeled engineered protein that binds integrin receptors into the vasculature, so tail vein injections of these mice. Mice with tumors light up. We just need to shave their heads to be able to remove the scatter that comes from their fur. Mice without tumors, no signal. And we see that in both the implanted and the genetic models. When you remove the brain tissue, the areas of clearly disturbed tissue also lights up. And we can quantitate this, right? So when you're doing molecular imaging with the molecular imaging equipment we have, we're able to measure the fluorescence that is from that tissue, and we can quantitate this. And both in these two different mouse models we have, when looking at mice with tumor compared to no tumor, and look at that total radiant efficiency, so how much light is coming from that area, we see those clear differences, right? To the extent where you really can separate mice with tumors from mice without tumors for both models. And so that's all in mouse, right? So that's all in mouse. So how do we start looking at what's our first application? How do we get that to actually helping our patients? So we can see the presence of the medulloblastoma in the mice, but what about in this patient? This gets into the aspects of surgical resection that many of you here are also looking at. And so that was our first application area we wanted to look at. So if we're doing optical imaging, we wouldn't realistically be doing optical imaging with this patient in this kind of not in the surgical suite. The light won't transfer through the skull of the human patient. And we're not really screening these populations, right? It's when the patient presents that they are sent in for an MRI, and that's actually pretty good at detecting a tumor at this stage. The patients who will have resection will have follow-up screens, so these molecules could potentially be converted into MRI probes to help detect any return earlier. But really our first application we're interested in, we're going after right now, is surgical resection of these tumors. So in trying to develop towards that application, we've collaborated with another group at Stanford that's developing a handheld confocal microscope for intraoperative use. So if you've used a confocal microscope, you know that you get really high resolution images. But what you're doing is you have this massive microscope, and you go into a room dedicated to it or dedicated to a couple other microscopes, and you're not able to do that with the child's brain when you're doing surgery, right? Has been developing, you see an early prototype here, they developed the optics to get this handheld version of the microscope so that a surgeon would be able to see at this fine scale tissue or if you don't. With the idea being that incorporated into the same probe, the same device, is this large field fluorescence too, if you just want that general big picture. Is there still tumor tissue or is there not? And then being able to switch back to, okay, am I at that margin? Because I don't want to leave any tumor tissue, but I want to make sure I leave all the healthy tissue I can possibly leave, right? And so then we tried this scope, this prototype, with our molecule to see how close are we to that margin. Are we able to delineate tumor tissue from healthy tissue? And in this case, we're looking at, we changed out our fluorophore. The prototype of this microscope was in the green range. So in this case, we had a mouse with a tumor. B should hopefully be an easy case, right? That should hopefully be all lit up, all tumor tissue. The microscope should be able to see that's all tumor tissue. C should hopefully be an easy case. This should hopefully be all healthy tissue, no fluorescence present in that section. And then if we're looking at the margin, labeled here as D, we should, hopefully, if it's working, be able to see tumor tissue in part of that section and an area lacking fluorescence in that section. What I'm going to show you here are examples of each of those sections. So in this case, the white dotted line is indicating where there is tumor tissue that we're looking at in this field. And in this case of tumor tissue, we do indeed see the entire area is fluorescent. And in that other, hopefully easy case, normal tissue, there is tissue there, but we don't see any fluorescence. And in each of the cases, as many of the margin cases that we tried, we do see this delineation. We do see part of the scope is lit up with fluorescence, part of it is dark, indicating healthy tissue. So that's where we are with this data right now. Again, some follow-up work has shown that, yes, we can repeat these with samples from human tissue. And we're not in people yet. And that is the direction that we want to take this. And there are some other collaborators who are doing work already with surgical resection of medulloblastoma, where doing all that additional preclinical work before you need to get to people for validating the ability of these proteins to go into people. That's a really first direction we want to go with this work next. So check back in with me as this work goes along, and we'll see how far we can take this. And that work has really opened up a few other lines of questions that I'm also continuing to work on, building on these questions of how do we engineer proteins so they can be used for cancer, both for diagnosis, like in this molecular imaging work, but also for therapy. And in particular, I'm interested in developing, you might have heard this nice little mashup word, theranostics. So molecules that can serve both the role of therapeutics and diagnostics. And the reason that we're interested in that is multifold. But one is when you're developing molecules for use in people, it's a long process and it's a steep cliff to fall for ones that don't actually get to implementation. So if you can develop a molecule that can serve both roles, then you're able to further patient care in a more rapid manner. But also, if you can pair these molecules, then you can do a better job identifying which patient, if they are diagnosed as having this molecule present, you should be able to give them that exact paired therapeutic and know they should respond to it, right? You're not giving them some variation or you think they'll respond as therapeutic. So it helps you identify which patients should get which therapeutic. And you can follow response to therapy. So if you have a therapeutic that's supposed to reduce the presence of a receptor or block the ability of a receptor to receive signal, and you can then use this paired diagnostic to, after you've been dosing this patient for a while, see if that's actually happening, then you have much earlier information as to whether you should keep on that treatment regimen or do you need to change over. And that lag time that clinicians currently get between when they know whether or not a patient is responding to a drug is currently much longer than it should be. And so this idea of these paired therapeutics and diagnostics has a real potential to try to shorten that information lag to help with better clinical care of these cancer patients. And the first set of cancers that I'm going after include some cancers that there just currently aren't good therapeutic targeted options for. Triple-negative breast cancer. So when thinking about triple-negative, cancers have different subtypes. They are often categorized, one way to categorize them is which receptors they have present. Some are estrogen receptor positive, some are HER2 receptor positive, some are progesterone receptor positive. Those are 3 receptors. If they don't have any of those, then they're negative. They're triple negative. It's not so much that triple-negative cancers are all one type of cancer. It's just that they're not those others. Those others have treatment options, but triple-negative has really poor prognosis and not treatment options. Ovarian cancer is usually detected very late, and there aren't good treatment options. Same thing with pancreatic, and some subsets of lung cancer also fall in that category. And so we're looking at specifically a tumor receptor pairing, actually. So we'd like to be able to see these tumors, right? Tumor receptor pairing, one's called mesothelin, and another one's called MUC16. What these 4 cancers have in common is a large percentage of them in clinical samples are turning out to express on their surface mesothelin, which interacts and binds with MUC16, both on, they're often expressed on the same cell, and they can also be expressed on having different cell types, so heterotypic cell interactions. And everything is not quite known about this interaction of mesothelin and MUC16. It's being actively studied, but it is one of these molecules that's turned on during development, off during healthy quiescent state, and back on during tumors. And if the ability of mesothelin to interact with MUC16 is interrupted, then that reduces metastasis and reduces spread and progression of these tumors. So going a little deeper, our strategy to engineering a protein that can serve as both a diagnostic and a therapeutic for mesothelin and MUC16 is to look at engineering proteins that sit in and block this binding interaction. So if this is the mesothelin protein that we're trying to engineer proteins to target, and this is MUC16. MUC16 is a mucin. It has a lot of glycan chains, highly glycosylated, a lot of sugar molecules, and those are what actually interacts with mesothelin. So if we're able to engineer a protein that binds very specific to this binding domain of mesothelin, which has been previously identified, then that molecule could first bind to mesothelin when injected into a patient to help with diagnosis. For some of these cancers, like ovarian cancer, mesothelin is also shed into the urine. so you can have earlier detection and screening if we're able to have molecules that can do in vitro diagnosis looking for this molecule. And if we have a molecule that blocks that interaction, then it has great potential as a therapeutic, since blocking the interaction is able to reduce metastasis and reduce progression of these tumors. And so the approach that we're taking is engineering. I pointed out this molecule earlier, this 10 kilodalton, the 100 amino acid protein, which is a very stable domain of human fibronectin. In its role of fibronectin, cells bind to it to help form tissue structures. In our case, just like in that cysteine knot protein earlier that had loops we could change without messing with the overall structure, there's loops that project structure that we can also mutate, and the sequences that we can mutate and swap out, we can mutate them to bind a new target. And the reason we're interested in this protein, and we started with this protein, is at this intermediate size that is able to clear rapidly from non-target tissue, but is able to stay retained in tumor tissue, which when thinking about we want clearance from non-target tissue so that we reduce background, but we want retention in normal tissue. And there's some of your colleagues here are working on other ways looking at this idea of getting contrast that I was really enjoyed hearing about today and I think are really promising. One of our ways of looking at this is engineering molecules that are able to be retained so we can wait 10 hours for non-targeted signal to clear out, so then we have retained tissue with the signal. It's also a very highly stable protein. There are variants of this protein that have been engineered that have melting temperatures that are well above what you would need for it to sit on a shelf as an in vitro diagnostic for a while without refrigeration. So I'm thinking again about in vitro diagnosis. Would we be able to detect in urine samples? That's one of the properties we're really interested in there. So just to remind you how this approach, so what we're doing to engineer this domain of fibronectin to bind mesothelin, we're using yeast cell surface display. So in this case, here's our yeast cell wall. We have protein variants, these variants of this fibronectin domain, tens of millions of variants of this fibronectin domain with different loop sequences. And we can incubate a library, so tens of millions of yeast cells with fluorescently tagged. The binding domain only of mesothelin is what we're working with here, and that's what we've expressed and purified. And then we have this library of tens of millions of different yeast cells, each one with a different copy of this fibronectin scaffold, incubate it with our fluorescently labeled mesothelin binding domain, that domain we want to block the interaction from. Use flow cytometry, identify those proteins that mediate binding and therefore fluorescence, and isolate clones that have this high, tight binding to our tumor target, but not just anywhere on the target, to that spot that could potentially interrupt that signaling cascade with MUC16. And we're in the middle of this process, but it's going in the right direction, right? So this is it's not a perfect process. It's not where you have tens of millions and you select one out the first time. There's a distribution of how much signal is there. So shown here are flow cytometry plots. On the x-axis is the protein is on the surface, so that's detected with this antibody to an epitope tag. And on the y-axis is binding to our tumor target. So what we want are molecules that are in this box, basically. So they express well and they bind well. Each one of these dots denotes a single yeast cell, which has a single copy of a protein variant on its surface. And so as we go through this process, we start to see this population, stable population emerging in this binds and expresses category. So that's where we want to go, right? So sometimes we'll do these protein engineering approaches, and no matter how many times you try to take this slice of cells, they keep returning back to when you grow them back up, they don't really have that stable expression. But we really are starting to see the cells come up here, and we've started looking at some of the sequences. And this is very early. These are not our end sequences. We'll be injecting into in vivo samples. But when you group them, you start to see patterns. And I just boxed what some of them are, just to draw your eye to them. And when you start to see those sequence patterns, that means there's something real going on there. Because you would expect that motifs would be emerging that are these binding motifs. And in this case, unlike for integrins, where we knew there was this tripeptide RGD binding sequence, it was like glycan chains, like glycosylation. Sugars interacted with this binding domain. Sugars aren't amino acids, so we didn't have a starting point. So that's why we started with this broad library. But now we're starting to get these sequences. And so we're continuing with this process. Directed evolution is iterative. My members of my lab are going through and adding in more mutations and selecting out more proteins until we start narrowing it down, where we have those nanomolar binders that start being interesting in clinical applications. So again, on this project, check back in with me. We're really excited the direction that it's going, and really looking at, if we have this molecule in hand, if we can develop that protein that doesn't exist in nature, all the different ways that we have the potential to interact with diagnosis and treatment of these tumors that express mesothelin that currently don't have options available. So hopefully I've convinced you that protein engineering is fun and cool and does great things in the ability to treat and detect human disease, in this case cancer, for what we're talking about here. But the aspects of protein engineering, and I know there's already some people who drunk the Kool-Aid here. We have some protein engineers in the room. I think it's just fascinating that you can make biological molecules that have these very specific activities that don't exist in nature. And really looking forward to continuing to pursue these lines of work, and I've been enjoying hearing what you all are doing with that today. And towards this goal, ultimately, of impacting patient care. I'd like to thank the people who are also involved with this work. I mentioned Melanie Hayden is the neurosurgeon collaborator that I've had on the medulloblastoma work. Fantastic person to work with, really dedicated to her patients, a great researcher, great collaborator. Steve Sensarn is developing right now, is one of the people developing that intraoperative confocal microscope. Did some of that pilot work looking at, well, can we really delineate tumor margins here? And these are the brave members of my lab at Smith College. So Fatima, Kadi, and Allison have been the three students doing most of the work with the mesothelin project that we've been initiating. And I have a variety of students from neuroscience and chemistry and biochemistry and engineering, which is a really great group of people to work with in my lab. So I'd also like to thank you for being here today and having a chance for me to share this work with you. And I'd be excited to take any questions that you may have. Right, so it's more, most of the work so far, as far as in vivo models, will be able to answer that question, have looked more at the metastasis. So it reduces the formation of new metastases and also reduces the spread of the tumor as far as tumor progression. I'm not familiar with any work that has yet asked that question of, do we deaggregate the tumor as far as in vivo aspects. Some of the in vitro work, looking at those cell-cell interactions, has shown that you do have in this monolayer, either when you have two different cell types, one expressing each of the receptors, or one cell that accepts both, somehow blocking maybe with an antibody that intersection does create this deadherence from other cells and subsequent cell death. But that bringing those together to really answer the question you're looking at would be that in vivo work looking at that deaggregation, and I'm not familiar with that question being asked yet. Yeah, Carl. Thank you.
Could you maybe just comment a little bit in more detail about how those monotonous cross the blood-brain barrier?
I can speculate. That isn't still working on, and so I can talk about some of the experiments we did to answer that question, and that goes into the work I didn't talk about. The fact that it's such an interesting open question relates to the work that I'm doing engineering proteins to be able to get past the blood-brain barrier and target various disease targets of the brain. So questions when you're looking at the blood-brain barrier that you start to ask is, well, for one, is the blood-brain barrier intact? So this, just briefly, the blood-brain barrier, there's very tight cell junctions in the blood vessels in the brain that regulates and protects the brain from harmful molecules getting in the brain, but it makes it really hard to get therapeutics in there. So the first question is, well, is the vasculature abnormal enough that molecules just leak across it? And so questions to try to it didn't include in this data here, but some of the experiments to try to answer that question was, one, injecting just tracer molecules at like dextran or other larger molecules and seeing, do they leak out of the vasculature? And surprisingly, the answer is they don't. So the vasculature is intact enough that these larger molecules don't just leak out. And so part of a challenge with that is these cysteine knot proteins are small, they're 3 kilodaltons. So maybe it's just small holes that they're able to leak out. Maybe it's not just the targeting aspect. So then we made a larger molecule, reformatted that cysteine knot protein as a fusion, an FC fusion. So made it basically 100 kilodaltons with the way we did the fusion. And so then it's larger close adduction, and that was still able to target the tumor tissue. So it wasn't just this fact of the molecule was small. And it wasn't just the fact of the vasculature is leaky. And those are things that we thought would be happening. But we're not sure, is it an active transport mechanism? Do integrins do something with that? Don't know. One thing that also could be, well, maybe we're only binding tumor vasculature. Maybe we're not actually binding the cell since the receptors were present on the vasculature on the cells. But when we look at the histology, and again, I didn't include this in here, that molecule is binding both the vasculature and the cells. So yeah, I don't know why that's getting crossed. I think that's really interesting. People have gone after other active receptor transport to try to get molecules into the brain. Transferrin receptor, for example, is a molecule that brings iron specifically into the brain. And that's how in drug design, some people are trying to design molecules that can hijack that system, basically hide as though they were transferrin and have the transferrin receptor pull them. Are integrin receptors able to do something? I don't know. I think really interesting questions, but not sure why it works. And try to answer that question and just don't have a very solid answer yet, which I think, again, is what really got me interested in that direction. Is I just think there's a lot of meat in there into that question.
So maybe just follow up.
Yeah.
You all had not finding other targets besides the integrin receptors. Is it known whether or not those penetrate the blood-brain barrier?
Yeah, so the fact there is a not one that we were working with, but one that is already, it's been in clinical studies, a non-engineered nautin, but one that natively exists. It's a conotoxin. So I think it comes from, is it a spider? It's just like a cone snail toxin or something. And it does naturally bind to some tumor tissue. Molecular and efforts for people to try to figure out why it binds to tumor tissue haven't been figured out. And so the fact that nautin bound to tumor tissue was one of these things that when thinking about, can we have this engineered nautin and binds to vasculature, what about brain tumors? Because that's a really hard problem, and nautin seem to get into brain tumors. That one does seem to have some sort of interaction, but again, any satisfactory really results that say, yes, it does cross it intact blood. It's just, the data hasn't, there's been attempts, but the data sometimes gives different answers, and I think it's a really interesting question.
But I guess, you're talking about those medulloblastic tumors, and they contrast enhance, so that shows that the gadolinium.
It does, right. Right, and so one of those, so it depends on what stage of the tumor is too, right? Like is gadolinium contrast leaking out later stage or early stage? And you don't usually catch the early stage enough to ask that question in a human. I did some, again, it's not here, but I did some work with MRI with these mice and then compared it to with various different sequences. And some of them contrasted, but not as well as we would expect based on the fluorescence we saw. And some of them didn't really contrast enhance. So again, that was all part of trying to get to this question about the blood-brain barrier. Does that explain what we're seeing? MRI with mice is hard. Anyone else has done it? With gadolinium with the mice, yeah. I saw some hands over here. Yeah.
So how long does it take for the engineering process? Like for years?
Oh, forcing, that's an it depends type of question. So how long does it take to engineer a new probe? So if you have this interesting target, which I'm always interested in new interesting receptors if you want to talk to me about your favorite receptor. So that molecule that I was talking about, the medulloblastoma work, is probably a process of 1 year to really get molecules that had clinically relevant binding. And that one was where there was a starting point of a binding sequence that could then be further engineered. But enough of the methods have been developed by the family of people doing directed evolution and protein engineering that a year to get to a molecule is reasonable as a starting point. Then you have projects like any project that you encounter every barrier you don't expect to encounter, and it's much longer than that. It's not an overnight process. As far as the rounds of like a set of directed evolution, like those plots that I showed you were collected over probably a month during which my student was doing that stage, and then the process of introducing new mutations and doing that round again. So several months on that stage, but then making the libraries, making the protein molecules that effluorescently label your target and purifying those, and then starting to characterize them and recovering the DNA sequences. That whole process is more of a year. Okay. Yes?
I have a question on the
Yes.
The images look very binary, either tumor or no tumor, which is what you want at the end of the day, obviously. What are the challenges in extracting the useful signal out of the
Right.
perhaps messy data that you have to work with?
Right. And so there's the qualitative data that where there's those images, and then there was the quantitative. I had some histograms up there, and those they had a cutoff, but there was a distribution around that. And so the binary aspect is when we show those images, we can say what is red and what is yellow, and set them where you can see that. If you set a really broad range, then it would seem less binary. And so your question gets more into the quantitative aspect of that. And so that's one of the things with molecular imaging, being able to pull out what is that total flux, what is that radiance through that area, and having enough. You would have to have enough data set where you have a sense of what is and isn't tumor. If you were doing diagnosis with it and you wanted that yes, no, our bonus that we get with doing surgical resection is there's tumor. And so it's, is this cell or is this cell not there? And so that's where we would have to work out, for example, washing procedures to make sure that it's not, there might be some nonspecific sticking nearby of those inflamed cells. And you want binary, but in practice it's not completely binary. And I think it's both in the data processing aspect of that. Again, some of the subtractive work with having some sort of a tracer that might say this is what the background just nonspecific sticking is. Those types of things can be helpful. It's a tough, you want that binary, and it's not there. It is tough. It's a hard problem and an important problem. You're just waving your hand. Anyone else?
This is a topic that is near and dear to many people's interest here, so I really would like to thank you for coming out.
Thank you. Great. Thanks for having me.